Okay, so in this video we're going to be looking at the vessels of the arterial system and the vessels of the capillary networks. So that's the plan, and then we're going to talk a little bit about the exchange of materials across the wall of the capillaries. All right, so we'll start by talking about arteries and arterioles. Again, this is just a reminder now, they conduct blood from the heart. So any blood vessel that is carrying, regardless of the kind of blood that's inside of it, the oxygenation of that blood, regardless if it's carrying blood away from the heart, then we refer to it as an artery. Sorry, it's late and I'm still doing these lectures, and so I'm going to try not to yawn, but there it is. So, arteries are composed of all three of the tissue layers. And we could probably remove that, not necessary. And the tunica media is the bulk of the artery wall. It's relatively thick in order to withstand the high blood pressure that arteries must endure. The larger arterioles also have all three of these layers, but the further that the arteriole is from the heart, the thinner the tunica media becomes. So by the time we get to the very smallest of arterioles, there may only be just a thin endothelial lining, that little single layer of endothelial tissue, some scattered smooth muscle tissues, kind of smooth muscle cells wrapping around the arteriole, and then just a little bit of a connective tissue sheath surrounding the vessel. So the further away from the heart, the less of everything around it, with the exception of the endothelial lining. That remains intact throughout the entire blood vessel system. So the capillaries, these are the smallest blood vessels. They connect the smallest arterioles to the smallest venules, and they're composed only of tunica interna. All that we see of a capillary is that tunica interna, the endothelial layer. And they can be found in these branched interconnected networks. So we see branching and then reconnection all throughout the capillary bed or the capillary network. And the density of the networks is really determined by how much oxygen the tissue needs, which is reflected by its metabolic rate. The higher the metabolic rate, the higher the need for oxygen in those tissues and nutrients, of course, to do that metabolic work. So the higher the density of the capillary networks. The lower the metabolic rate, then the lower the density of the capillary network. And if you recall, we talked about cartilage as having relatively little blood supply. And there are some tissues that have no blood supply at all. Things like the cornea, the clear layer at the anterior surface of the eye. So in between the endothelial cells that make up, and the book has a great cartoon of this, in between those endothelial cells there are these gaps, spaces where there is no cell, and these are very small little spaces, very tiny, and they're referred to as intercellular channels, so that they are little channels between the endothelial cells that make up the capillary wall. And it's the size of that channel that controls how permeable the capillary is. So the larger the intercellular channel, then the greater the permeability of the capillary. So we see larger intercellular channels in capillary networks that are near to endocrine glands, the ones that are producing the hormones that need to be picked up and carried by the blood. They're larger in the capillaries of the kidneys, where we're busy trying to get all of the waste cleared out of the blood, and larger in the capillaries near the small intestine, because that's where we're picking up all the goods from the small intestine, the digested bits of fats and sugars and proteins and so on. So the entrance to each of the capillary networks is controlled. So if this is that network here where we have these interconnections between the capillaries of the network, there's an entrance controller here that's called the precapillary sphincter. So it's precapillary because it's right before that capillary network, and it's a sphincter because it's composed of smooth muscle that wraps around the blood vessel. So sphincters are always smooth muscle, and they wrap around a tube of some sort. So there's the anal sphincter, there's the sphincter between the stomach and the duodenum, the first little part of the small intestine. So these are all built so that the muscle fibers wrap around it like this, so that just like with blood vessels in any sphincter, when the smooth muscle contracts, it's going to pinch off that the flow of blood or whatever, poo or food, through that particular region, that opening region. And these precapillary sphincters, they respond to changes in oxygen and nutrients. So if here in the tissues, so we've got cells around here, if amongst all these cells, the interstitial tissue is really low in oxygen or in important nutrients for energy, then the precapillary network the precapillary sphincter will open, allowing blood to come into the network, bringing with it, hopefully, some oxygen and some nutrition, some food that the organism can, that the tissues, cells around that precapillary sphincter can use as an energy supply. So the routing of blood flow through the body is really controlled by the vasoconstriction or dilation in arteries and arterioles, and then that contraction and relaxation of precapillary sphincters. When this region is got plenty of oxygen and plenty of nutrients, the sphincter is going to close, and the blood then will be routed over toward this particular precapillary network. So rather than sending the blood to the tissues that already have lots of oxygen, no, we're going to instead send it over here to the tissues that have an open sphincter and a need for oxygen or for the nutrients that are found in blood. Okay, does that make sense then? It's for the flow of blood through a capillary network, it is all going to relate to local tissue conditions. If the local conditions are good, the sphincter will close. If the local conditions are not so good, if we need oxygen or need food, then the sphincter will open. So exchange across the capillary occurs by processes that we've already discussed in this course, actually last semester. Diffusion is one of those processes. So out there in the systemic circulation, blood entering capillaries is going to be high in oxygen and in nutrients because it's been to the lungs and now it's going out to the tissues. And diffusion will move, so if this is capillary network here, or capillary, and here's the blood coming into that capillary, it's going to be high O2 in this blood. Out here in the tissues, we're using up oxygen. So it's lowest O2 in here, and in between it's going to be kind of moderate O2. So the oxygen is always going to move from the region of highest concentration out here to the region of moderate concentration, and then from there into this region of very low concentration, all by diffusion. Now at the same time, carbon dioxide is being made inside the cell because the mitochondria are using the oxygen and the glucose to make energy, or make ATP, the cellular energy. So this is going to be high CO2 here inside the cell. It's going to be kind of moderate CO2 in the tissue fluids, and here it's going to, as it leaves the capillary network, because of the exchange that's occurred, this is going to be because of the exchange that's occurred in the lungs, this blood is going to be very low in carbon dioxide. So the carbon dioxide is going to move from the cell into the tissue fluids and then on into the blood. So diffusion moves most of the gases and a lot of the nutrients. Filtration can also occur. So here in the on the arterial side of a capillary network, blood pressure is going to push fluid out of the blood. So we'll see a lot of fluid being pushed out of the blood. Here at this end of the capillary network, though, with all these proteins in the blood, and they're going to be even more concentrated since we've squeezed out a lot of the fluid, well, the proteins are going to be in high concentration here, and so water is going to move by osmosis from one side of the capillary wall to the other side of the capillary wall. So oxygen and carbon dioxide diffuse down their concentration gradients, and water will diffuse back into the blood down its concentration gradient. So at the arterial side of the capillary network, what we're going to see is plasma that is rich in oxygen is going to be forced from the capillary and into the interstitial fluid. It's going to carry with it all the digested triglycerides and the digested mono and disaccharides and the digested amino acids out there into the interstitial fluid. So that's here at the arterial side. So if we're looking at, here's an arterial, and we're heading into a capillary network, and then we're going out back toward the heart. So in comes the blood, here out goes the blood, and this region in between the arterial and the venule is all going to be the capillary network. So here at the arterial side of the capillary network, blood pressure pushes fluid out because the blood pressure here is so, in the, oh shoot, I should have my book with me and I don't. I believe it's like 34 millimeters mercury. One moment, I'll be right back. Sorry about this. I'm used to having this conversation with the book here in front of me. So if you look on page 357 in the textbook, that can't be right. Yeah. Sorry, wrong textbook. Page number. Okay, here we are. In here. All right. So this is figure 1530 on page 581. So yeah, there's a force of blood pressure pushing fluid out of the capillary with about 35 millimeters mercury blood pressure force. Now, that's going to be offset some by the osmotic pressure calling fluid in because of all of the proteins, the plasma proteins that are found in the blood. So we've got a pressure out of 35 millimeters mercury, a pressure inward of 24 millimeters mercury. So we have to do a little subtraction problem. And what we see is that overall, we have about 11 millimeters mercury force pushing blood plasma out of the capillaries on the arterial side. Now, here at the venous side, on the other hand, we have a blood pressure has really dropped. It's only 16 millimeters mercury, but we still have that 24 millimeters mercury draw into the blood. So overall, we're going to see a -8, which means that there is about 8 millimeters mercury force pulling fluid back into the blood here on the venous side. So because of the higher blood pressure here at the arterial side, what we see is that plasma gets forced out into the interstitial fluid, carrying with it oxygen and nutrients and hormones. All sorts of stuff gets carried out if it can pass through those intercellular channels. The rest of it must diffuse across the endothelium of the capillary or be actively transported out of the blood or back into the blood. Here at the venous side, the interstitial fluid is drawn back into the capillary, bringing with it carbon dioxide and other wastes of metabolism, and anything that the tissues are trying to get rid of gets put into the blood here and then carried back by the venous circulation back toward the heart. And what we see is that generally more fluid leaves the capillary than returns to the capillary. And so that's where the lymphatic system is really important in collecting that extra fluid that didn't make its way back into the capillary and returning it back to the blood circulation. And so when we get to the next chapter, that's all about the lymphatic system. Then we'll be talking about how we do that kind of work as well. Okay, so hopefully that all made sense, that between all three of these processes of osmosis, of diffusion, and filtration, we're going to see the push out of oxygen and nutrients and hormones, and then the drawback in of fluid and waste materials like carbon dioxide and other metabolic wastes. Okay, so now we're back to the blood vessels again. We've made it to the far side of the capillary networks, and so the next blood vessel that we see are the smallest of the veins, the venules, and they just resemble a really enlarged capillary, but as the venules continue to merge, then they begin to pick up the characteristics that we associate with a full-on vein. Tunica media becomes thicker and thicker as more and more smooth muscle cells are found in that tunica media layer, and a connective tissue wrapping around the whole blood vessel also begins to appear, and so then we have the tunica externa. So eventually the venules merge enough and become large enough that we refer to them as veins. Veins have all three of these blood vessel layers, but the blood vessel wall is relatively thin compared to that of an artery because the tunica media is relatively thin. So less muscle, less elastic tissue than we'll find in comparable arteries in the same region of the body. As well, the lumen, that's the interior of the vein, is larger than the lumen of a comparable artery. So lots more space inside that vein for the blood to hang out. So what we see is that as blood leaves the capillary networks, blood pressure is now down to about 16 millimeters mercury. And so the blood pressure in the veins becomes lower and lower as more and more venules hook together, creating larger and larger veins. And so in some veins, there isn't sufficient pressure to overcome the force of gravity if you're standing up. And so in the limbs, especially, the veins have one-way valves that prevent the backflow of blood. So if this is a vein running up the leg, let's say, there will be these one-way valves kind of like this, similar in construction to, say, the pulmonary valve or the aortic valve. And so as blood is moved back toward the heart, some blood gets squeezed into this this section of the vein, and then as the heart stops beating for a brief second, then that blood will kind of fall back down with gravity, and that will close up the valve. And then later on, a little bit more blood gets forced into this one, forcing blood on up into the next section. And then when the heart is in between beats again, then the valve snaps shut. And so, you could imagine that in this way, blood gets all the way up our long legs and our long arms as it's heading back toward the heart. And if the walls of the veins near these valves will stretch, then the blood will pool. So here's an area where, say, there's been some stretching that's occurred, and so the blood can wind up pooling around that vein area. And we refer to these as varicosities, so varicose veins, and as hemorrhoids if they're found there at the rectum, especially right at the opening of the rectum to the external environment. Okay, so there are the venules and veins, and that is it for this video. So again, to remind you where we've been, we've talked about the wall of and the construction of the arteries, of the arterioles, of capillaries, of venules, and then of veins. And we've talked a little bit about the processes that allow for exchange between the blood and the interstitial fluids, primarily osmosis and hydrostatic pressure, causing filtration and diffusion of materials along their concentration gradients. So there you have it. Only two more lectures left, and don't hesitate to make contact with me by email or phone call if you have any questions about this material.